Welcome back to Summarize Science. Today, we're diving into the world of microproteins, a newly discovered class of incredibly small proteins. Think of them as the tiny, specialized tools in our body's cellular toolkit. Just like a real-world tool, a protein's 3D shape determines its job or function. The problem is, because these microproteins are so small, it's extremely difficult for scientists to see their shape using traditional lab methods. This is where computers and artificial intelligence come in. In this paper, researchers set out to see how well our best computational tools could predict the 3D structures of these tiny molecules. They selected 44 different microproteins and ran their sequences through 3 of the most powerful prediction programs available: iTaser, Rosetta, and the famous AI, AlphaFold. The first major test was to compare the computer predictions to the real, experimentally known structures for 3 of the microproteins. They found that the AI, AlphaFold, gave the closest predictions to the real thing, which is what you might expect. However, the predictions were not nearly as accurate as AlphaFold's predictions for larger, more common proteins. It's like an expert artist being able to perfectly paint a large mural, but struggling a bit when asked to paint on a tiny grain of rice. Next, the researchers compared the 3 computer programs against each other to see if they would at least agree. What they discovered was fascinating. For many microproteins, all 3 methods agreed on the central core of the structure, which was often a simple helix shape. You can see that here with sarcopene, where the different colored predictions are all coiled together quite nicely. But when it came to the floppy ends of the proteins, the predictions were often completely different, going off in all directions. This other example, the negative regulator of Peabody, shows how much disagreement there can be when there isn't a stable core. There was also an interesting twist when they measured the compactness of the predicted shapes. In this specific test, the other 2 programs, iTaser and Rosetta, actually produced results that were more in line with the experimental data than the powerful AlphaFold AI. This shows that each prediction method has its own unique strengths and weaknesses when dealing with these unusual proteins. So, let's recap the important points from this study. First, our best computer models can give us a general idea of a microprotein's shape, especially its central core. Second, these models, including the powerful AI AlphaFold, are not yet as reliable for these tiny proteins as they are for larger ones. And third, the models often disagree on the flexible ends of the proteins, which could be very important for their function. What does this all mean for the future? The researchers conclude that the biggest hurdle is a lack of real-world data. The AI and other programs simply haven't seen enough examples of verified microprotein structures to learn from them effectively. The solution is for scientists to focus on determining more of these structures in the lab, creating a bigger library of examples. With more data to train on, these powerful prediction tools can be fine-tuned. This will ultimately help us understand the roles of thousands of these mysterious microproteins in our health and in disease. Thanks for watching, and be sure to subscribe for more Summarize Science.